For the second part of our discussion on the central dogma, we will be talking about transcription. And we finished our discussion on DNA replication. So we are finished with this part of the central dogma. So now we are focusing on the creation of the mRNA from a certain DNA sequence. So that is the process that we call transcription. So now this process, remember we said that DNA replication takes place in actively dividing cells, but transcription takes place continuously inside cells because this is the process, an intermediate process in the creation of the gene product, which is your protein. So any live cell will be continuously performing transcription as long as it needs the protein product of any genes that should be expressed. Okay, so we already know that your RNA molecules are more diverse. So these are single-stranded nucleotides made up of your four nitrogenous bases, your adenine, uracil, cytosine, and guanine. Now we will be focusing on the mRNA. So here in this chart, we see that we will be focusing on this type of RNA, but keep in mind that the process, the creation of the other types of RNAs indicated here is the same, okay? But again, since we are after the gene product, which is the protein, hence our focus is on the mRNA. Okay, so let's define a few terms. So again, transcription is the synthesis of an RNA molecule using a DNA template. Okay, and then when we talk about transcriptome, so transcriptome, much like when we talk about the entire genes of an organism, we call it a genome. The transcriptome is the entire set, the set of entire RNA transcripts being produced at any point in time by a cell. Okay, so it's the collection of RNA molecules, or what we call RNA transcripts, being produced at any point in time. So this changes based on the state of the cell. So your messenger RNA is the most of the RNAs produced via transcription are messenger RNAs. But again, keep in mind that the same process also produces other types of RNAs, such as your ribosomal RNA, your tRNAs, and of course, the smaller RNAs, which have regulatory functions. And then finally, we have RNA polymerase. So RNA polymerase is the enzyme that catalyzes the synthesis of your RNA molecules. Now, let's try to compare replication versus transcription because interestingly, there are some things similar between the two, and of course, there are stuff that are different. So number one, of course, is the enzyme used in each process. So for DNA replication, we know that the enzyme used is called DNA polymerase, while in RNA transcription, of course, it's an RNA polymerase. Now, for the direction of synthesis, it's the same for both. Recall that in replication, a new DNA strand is synthesized from the 5' to the 3' end, 5' to 3'. Similarly, any RNA strand that is being synthesized during transcription is also synthesized from its 5' end to its 3' end. So the direction of synthesis is similar. Hence, there should be some similarities in how the DNA polymerase and your RNA polymerase works, as we shall see later on when we look at the reaction mechanism. Now, for the template required, for replication, as mentioned, it requires a DNA template, which is also the same with transcription. So these two processes requires a DNA template as well for RNA transcription, but it only uses one of the strands in your double-stranded DNA. And the specific strand that it uses as template to synthesize the RNA molecule is what we call the template strand or the non-coding strand. So we will understand this a bit later. Okay. Now for the primer, we learned that when a DNA reproduces or makes an exact copy of itself, it requires primers. So primers required. This is synthesized by an enzyme called primase, which is part of your replisome. In the case of transcription, if this cell tries to synthesize an mRNA, it doesn't require a primer. So there is no analogous enzyme for primase in the process of transcription. So it simply starts with the first ribonucleoside triphosphate and so on. Now, as for the stages, the two are similar, which has the initiation stage, elongation, elongation stage, and termination stage. So initiation, elongation, and termination. But of course, the molecules and other enzymes involved in each stage would be different for the two processes. Now let's talk about the error rate. We've seen how the prokaryotic replication machinery is a very precise, so to speak. So it encounters 1 in 1 raised to 9 to 1 raised to 10 nucleotides. So that's the error rate for DNA replication. That's the average. Okay. Meanwhile, for transcription, the error rate is actually higher. So it's 1 in 1 raised to, is it? No, sorry, 10. 10 raised to 4 to 10 raised to 5 nucleotides. Okay, so let me just correct this. So for replication, it's 1 in 10 raised to 9 to 10 raised to 10 nucleotides. As we can see, there is a higher error rate during transcription. Why is that? So keep in mind that the objective of replication is to create an exact copy of your DNA, and the information encoded in your DNA molecule is very important. Any mistakes that will occur during one cycle of replication will be transferred to the subsequent offsprings, right? So the cell must ensure that it minimizes as much as possible the mistakes that can happen during replication. On the other hand, transcription has more room for error because it's a dynamic process, meaning at a time when you need a specific protein, that is the only time when you will transcribe that gene, right? And once you are done with the protein, once the protein is done with its function, the cell will eventually degrade it. And when you transcribe, the concentration of the RNA molecules, the product, the RNA molecules that are produced from your transcription process, they will not stay in the molecule for a long period of time. So once you produce the mRNA, it will eventually proceed to the translation process. It will proceed to the synthesis of the corresponding protein product. And after that, the mRNA will be degraded. And this will continue. You continue to produce the mRNA for the same gene. And once you reach a certain concentration of the needed protein, then the transcription stops. And once the protein molecules are done with their function, then they are also degraded. So it's a more dynamic process, and it is continuously being regulated, transcription, versus replication, which again only takes place if your cell will be reproducing or will be actively dividing. So this table summarizes the differences and similarities between replication and transcription. Okay, so now let us take a look at the architecture of the main architecture involved in transcription. So you need to produce an RNA molecule using a single-stranded DNA as the template. As we can see here, much like in replication, we also have what we call a transcription bubble. So the transcription bubble, you have a segment of your double-stranded DNA which is unwinded, and then your RNA polymerase reads only one of the DNA strands. So it only uses one strand of DNA as the template, and this is what we call the template strand or the non-coding strand. The template strand is also called the non-coding strand. So this is the one that is used as template for the sequence of the RNA molecule that is being synthesized. And as you can see in this architecture, we have a so-called RNA DNA hybrid, which is around 8 base pairs long. So as the RNA polymerase synthesizes this RNA strand, a certain segment of the RNA molecule keeps its interaction with the template strand, with the DNA strand. And this is again to maximize the stability of the whole complex. Okay, and then the direction again is from 5' to 3'. So in this case, if you can notice, the template strand, the directionality of your template strand is from 3' to 5'. Okay, so we don't have a term such as transcription fork in this case. Okay, so we only have what we call a transcription bubble. And finally, the other DNA strand that is not used as the template is what we call the non-template strand, or this is the coding strand. It is called the coding strand because the sequence of your coding strand is the same as the sequence of the RNA molecule being synthesized. The only difference is that the T, the thymines there, are replaced with uracils. Okay, now let's look at the mechanism of your RNA polymerase. And again, if you can notice, it's the same figure. Well, it's not exactly the same figure as what you've seen in DNA replication, because since this is RNA, there should be those OH groups at the 2' carbon. Okay, but the active site is, it looks similar. So you have magnesium ions and you have negatively charged amino acid residues in the active site that stabilizes the magnesium ions. These magnesium ions interact with the negative charges of the phosphates. And again, it's a similar nucleophilic attack of this growing strand of RNA. So the 3' OH here, which is now deprotonated, attacks the alpha phosphate. and then some electron rearrangement, and then a diphosphate, or this is the more correct term for this one is pyrophosphate, is released. Okay, and now you have an additional ribonucleoside or ribonucleotide in this RNA strand. Okay, so another thing that we can consider in the architecture of your transcription is the topological stress again. So if you recall, we have topoisomerases involved in relieving topological stress of your DNA during replication. So as the helicases unwind the double-stranded DNA and the replication fork moves, you have topoisomerases that relieve the topological stress of your DNA molecules. There is also topological stress introduced in transcription. So we have positive topological stress in front of your transcription bubble, and there is negative topological stress um, behind your transcription bubble. So I'm referring to the direction of the transcription. So moving forward, there are positive supercoils introduced to relieve the topological stress, and there are negative supercoils made behind the transcription bubble to relieve the topological stress. And again, these positive supercoils and negative supercoils are made possible through your topoisomerases. And I forgot to mention, since I mentioned helicase in the context of DNA replication, for transcription, so the unwinding of the DNA is not performed by a helicase. So we will be looking into that when we go to the stages of transcription. Okay, so remember, transcription produces an RNA transcript from a DNA template. It uses only one of the two strands of a DNA molecule as the template strand. The other strand is what we call the coding strand, because the sequence of your coding strand is similar to that of your RNA transcript, with only the T's getting replaced by U's. Okay? Now, you might wonder, so does it mean that it's always the 3' to 5' strand that is used as a template? No. The cell or the coding strand can be either of the two. So for example, here is a diagram of the coding information in the adenovirus genome. So it's composed of 3.6 times 10 raised to 4 base pairs. So the two strands are represented by the blue lines. And as you can notice, the genes are represented or indicated by the green arrows, which are the RNA transcripts from each corresponding gene. So here we can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 arrows, meaning there are 11 genes here. There are 11 RNA transcripts that can be produced from this genome. So there are 11. So that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And as you can notice, it's not limited to one strand of your DNA. So for example, for this RNA transcript, it will use this strand of the DNA as the template. Therefore, for this RNA transcript, its coding strand is this one. Okay? Vice versa, if we are looking at this RNA transcript, its template is found on the other strand of the DNA, and its coding strand is the complementary one. Okay? However, if we are reading from the other direction, as you can notice, there is an overlap. You can also get an RNA transcript from this segment of your DNA, but you will be reading on the second strand. So this is the template that you will be reading to create this RNA transcript. And then for this one, the coding strand will be the one here. Okay? So again, it's not limited to one DNA strand. It can be either of the two. Okay, so let's just take a look at the structure of your RNA polymerase, and this is for bacteria. So prokaryotic RNA polymerase. And then while we will not be limiting our discussion to prokaryotic transcription, we will be starting with it, and then we will be later on we will be talking a bit about eukaryotic transcription. So the structure of your RNA polymerase is a simpler one than the DNA polymerase, which, if you recall, for prokaryotic DNA polymerase 3, it has a lot of subunits. But for RNA polymerase, and again, this is for bacteria, this is the core RNA polymerase, and it only has 5 subunits. You have the alpha, beta, beta prime, and omega. So it's shaped like a glove, right? If you can notice. And then, since we're talking about the core, it has an additional subunit, but this one, the additional subunit is more of a modular subunit. It's not part of the core RNA polymerase. So the sigma, so this sigma subunit is modular. Okay, so there are several types of sigma subunits, and the core RNA polymerase can attach to any of these sigma subunits. Okay, the sigma subunits, as we shall see later on, dictates which gene gets to be transcribed. Okay, as long as that delta subunit, sigma subunit is present in high amount in your cell, especially in the nucleus, then, for example, sigma 32 is a special sigma subunit for the expression of heat shock genes. So if the bacteria is suddenly exposed to high temperatures, it needs to express these heat shock genes to protect itself, to survive the extreme high temperature. So it will increase the amount of sigma 32 subunits in the nucleus, and then this sigma 32 will be the ones to recognize these heat shock genes in the DNA of the organism. So it will bind to the core RNA polymerase, and then it will search for the specific heat shock genes that need to be expressed. Okay, so there are 7 commonly known sigma subunits for E. coli, but of course, there are others. And when it comes to the eukaryotic ones, then it's more complex. Okay, so now the question: how does the sigma subunit, to be more specific, identify the gene? Right. So remember, it's the identity of the sigma subunit that dictates the genes it can express. Okay, so remember, the sigma subunit binds to the core RNA polymerase, and the sigma subunit can recognize specific sequences that we call promoters for the specific genes that it's assigned to. Okay, so generally speaking, we have these so-called promoters. So these promoters found in the coding strand yeah, or in the non-template strand are recognized by your RNA polymerase via the sigma subunit. Okay, so as you can notice, there are numbers here like -35, -10, and +1. So this is a convention used to specify the location of your sequences in a strand in a nucleic acid strand. Okay, so for the nucleotide that corresponds to the first nucleotide of the RNA to be synthesized, we designate that as +1. Okay. And then going upstream. So when we say upstream is going more positive. So the next nucleotide is +2, +3, +4, and so on. And again, we call it upstream. Now, to the left of the +1 nucleotide, we assign it a number of or a label -1. Okay, to the left, the nucleotide to the left of the first nucleotide corresponding to the first nucleotide of your RNA transcript, that's +1. To the left, that's -1. So there's no zero. And then going to the left, we call that downstream. Wait, I think I got it opposite. Okay, so as you go more positive, that should be downstream. And as you go more negative, that's upstream. Okay. So more positive downstream and more negative upstream. So these terms and these numbers are again are used to locate specific sequences. Your promoters are located upstream of your gene. Okay? They are not exactly part of the gene because their sequence is not part of the sequence of the RNA that will be synthesized. Okay, so upstream you will see certain promoter regions, and we have here a -10 region, a -35 region, and we have a UP element. Okay, so UP, if I'm not mistaken, UP here stands for upstream element. Again, these are sequences that are recognized by your RNA polymerase to signal that somewhere nearby is the start of your gene. Okay. Now, the upstream element is more how can I say, the -35 region and the -10 region are more common. But the upstream element can only be found in some genes. Again, this is in the context of prokaryotes. Okay, so as we can see here in the caption, the consensus sequence for E. coli promoters recognized by Delta 70 is shown second from the top. Okay, that's it. So we have promoter sequences, upstream element, -35 region, and -10 region. The only thing now that we have to observe is that these promoter sequences are rich in A and T. A and T. So that is similar to what we learned in replication, wherein your origins, replication origins, they are typically rich in AT as well. Because again, AT base pair, is easier to break than GC. Okay, so now let's look at the stages of transcription. We will start with initiation. So for the initiation, first is your sigma subunit should be bound to the core RNA polymerase, and then it will try to scan the length of the DNA molecule for the promoter sequences. And once it recognizes the promoter sequences, like the -10 and the -35 region, it positions itself and creates this closed complex. Now, this is still the closed complex because the DNA is still um, it's still double helical, so it's not yet unwinded. So the RNA polymerase itself, one of the subunits of the RNA polymerase, is the one that catalyzes the unwinding of the DNA molecule. Okay, so a 12 to 15 base pair region of DNA from within the -10 region to positions +2 or +3 is then unwound to form an open complex. Okay, so if it is an open complex, that is the signal for the initiation. So here, the gene or the segment of the DNA where the transcription should start is now unwinded or unwound. Okay, then transcription can now begin. So other small molecules can also be involved in the process. And at some point, after what we call a promoter clearance, the sigma subunit will now detach itself from the core RNA polymerase. And that's when elongation will commence. Okay, so when we say promoter clearance, it means the RNA polymerase has moved past the promoter sequences. So it means that the RNA polymerase is now committed to finishing this whole process of transcribing that specific gene. Okay, so to be more detailed, to give us a more detailed picture of what is happening, we now have the core RNA polymerase here. And then we have the sigma subunit. To be more specific, what is depicted here is the sigma 70 subunit. These two will bind, form a complex with the sigma subunit recognizing the promoter sequences. And then the closed complex is formed. And then the RNA polymerase, one of the subunits of your RNA polymerase, will start unwinding the double-stranded DNA. And now you have an open complex and you now have a transcription bubble. Now, with the initiation of transcription, the RNA polymerase will now start to synthesize a short segment of the RNA transcript, and it moves slowly away from the promoter sequence. So once it clears the promoter sequence, the sigma subunit can now detach from the core RNA polymerase, and then elongation is now taking place. And then a protein molecule called NUS-A will bind to the same binding site of your sigma subunit. So the sigma subunit is actually replaced by another protein molecule, NUS-A. So NUS-A sort of acts like a termination factor. So as long as it's present there in the binding site of your sigma subunit, the RNA polymerase should continue transcribing. So it should continue synthesizing the RNA transcript. Now, if the transcription is terminated, the NUS-A dissociates, and then the RNA polymerase can now be used for another round of transcription. It can be the same sigma 70 subunit that can attach to it, or it can be a different sigma subunit. Now, the NUS-A The nose A is just there to fill the binding site of the sigma 70 subunit. It's not exactly a signal for the RNA polymerase to stop the process of transcription. Okay, so since we are now talking about termination, let's look at the two mechanisms by which transcription is terminated. So we have the raw dependent termination and raw independent termination. Raw here is a protein molecule which serves as the real termination factor for transcription. So let's look first at raw independent termination. So independent meaning it stops transcription without any termination factor such as the raw protein. So in this case, your RNA polymerase, once it reaches a terminator sequence, and this terminator sequence is typically A, rich in A. Okay, so it's continuous sequence of A's there in your template strand. A hairpin is now formed at the palindromic sequence that is upstream of the terminator sequence or upstream to the 3' end of the RNA transcript. Okay, so this hairpin disrupts the RNA DNA hybrid in the transcription bubble. So it destabilizes the RNA DNA hybrid. And because of this destabilization, it forces the RNA transcript to dissociate from the complex completely, and hence transcription gets terminated. Okay, so this is for the raw independent termination. So the signal for the transcription to stop is located in the sequence of the RNA transcript itself. Because due to the sequence of the RNA transcript, it tends to form this hairpin structure. For the raw dependent termination, we have what we call a raw helicase. So it's a helicase protein. It's analogous to the helicase that we saw in DNA replication. Okay, but its function is to separate the RNA transcript from the DNA template. So during the elongation step, there is called a RUT site or RUT there upstream. So it's near the 5' end of your RNA transcript, and this is recognized by your raw protein. So it will bind to this RUT site, and the raw protein will use energy from ATP to migrate along the RNA transcript. Okay, so it migrates along the elongating RNA transcript, and once it reaches the RNA polymerase, then the helicase will now catalyze the separation of the RNA transcript from the transcription bubble. Okay, so the timing at which the transcription stops has something to do with the migration rate of this raw protein. Now, aside from the dependence on the migration rate of the raw protein, the mechanism of this raw dependent termination also depends on whether your RNA polymerase has encountered a pause. Okay, so it encounters a sort of termination sequence in the template strand, so it stops. Okay, it stops, but termination is still not taking place. It just pauses Okay, the termination happens when the helicase reaches the transcription complex. Okay, so it's the migration rate of the helicase or the raw protein and the pause of your RNA polymerase once it encounters a certain sequence that allows it to pause or to stop. Okay, so that is raw dependent termination. Okay, so that is for prokaryotic transcription. Okay, now for eukaryotic transcription, it's a bit more complicated. Okay, and one, there are three RNA polymerases known to exist in eukaryotes versus the single RNA polymerase that we have for prokaryotes. So you have RNA polymerase 1, which is for the synthesis of pre-ribosomal RNA molecules. So it's dedicated to the production of ribosomal RNA. Likewise, your RNA polymerase 3 is dedicated to the production of tRNA, some ribosomal RNAs, and other specialized RNAs. The one that produces messenger RNA is the RNA polymerase 2. So this is for the DNA-dependent synthesis of mRNA. Okay, so just like prokaryotic transcription, we also have promoter sequences, but the position is a bit different. Okay, so we have negative 30 promoter sequence, and this is called the Tata box because the consensus sequence contains TATA sequence. Now, the sequence may vary as long as it's close as long as it's close to TATA, but it could be TAAA, it could be AATA, and so on. But again, we see that the Tata box is AT rich, just like the negative 35 and negative 10 regions that we saw in the prokaryotic promoter sequences. Then we have INR here, which is another promoter sequence. It's more of an initiator sequence, actually. So INR is for initiator. It's an initiator sequence, part of which overlaps with the RNA transcript. So the positive one position is found within the initiator sequence, but that is also recognized by the RNA polymerase. And then finally, upstream to the Tata box are various regulatory sequences. So it depends on the type of gene that is being that is located downstream. Okay, so the only common thing for eukaryotic transcription would be the Tata promoter sequence, which is located at the negative 30 position. Okay, so again, since this is a eukaryotic context, it's more complex. So there are more proteins involved in the process. And to be more specific, these are what we call transcription factors. Okay, and we also have transcription binding proteins or TBPs. These TBPs are the ones that recognize the promoter sequences. Okay, TBP or Tata binding protein. So the Tata binding protein, along with your transcription factors such as TF2F, which is bound to your RNA polymerase 2, your TF2B, TF2H, and TF2E, assemble in the vicinity of the promoter sequences. And if you have the initiator sequence there, so the TBP or Tata binding protein binds to the Tata region, which is upstream of your start position. And then you have other transcription factors assembled around your RNA polymerase. And if you notice, the initiator sequence here is recognized by the RNA polymerase itself. And since we are also comparing prokaryotic and eukaryotic transcription, I forgot to mention that your RNA polymerase 2 has more subunits, of course, than your prokaryotic RNA polymerase. Okay, so back to the process. So, binding takes place. Initiation is not yet achieved at this point. So, we still have the closed complex. Again, your RNA polymerase unwinds the DNA, the double-stranded DNA, and it knows where to unwind because it's interacting with the initiator sequence. So the transcription bubble forms. We now have an open complex. You have some transcription factors dissociating from the complex. So their work is done because their primary function is to help in the assembly of the transcription, uh, yeah, formation of the transcription assembly. You still have TF2F. TF2F is the transcription factor that remains bound to the RNA polymerase. Okay, so your once you have the open complex, initiation is now achieved. So transcription initiates. And then at this point, we have here a C-terminal domain of your RNA polymerase that gets phosphorylated. And this phosphorylation is important, as we will see in a bit. Okay, so it gets phosphorylated. There are several serine amino acid residues. So you have these serine residues have alcohols, right? So those alcohols, the hydrogen is replaced with the phosphate. So they get phosphorylated. And then the RNA transcript is now being synthesized. You now get promoter clearance. But as you can see, even though you have promoter clearance and you now have the elongation complex, the TBP there, the Tata binding protein, is still there, bound to the Tata box, but it's dissociated from the RNA polymerase. Okay? So once the elongation complex is formed, elongation factors become part of the transcription assembly. So they bind to the RNA polymerase. And once some signal is encountered, or no, to be more specific, once a termination sequence is encountered, so elongation is finished. So we now reach the termination step. So elongation factors are removed, so they dissociate from the RNA polymerase, and the CTD or the C-terminal domain gets dephosphorylated. So you see there the phosphates getting removed, and termination factors now become part of the complex. Okay, so all of these processes help in the release of your RNA transcript from the RNA polymerase. Now, you might be wondering, what are the functions of these elongation factors and termination factors? So, simply put, these protein molecules help in the stabilization of the complex. Some of them might have some proofreading capability, but very limited probably. But yeah, their main function is for stabilization of the complex. Meanwhile, termination factors, their function is to remove. So we don't want the complex to be stable at this point because you are terminating the process. So they aid in the removal of the RNA transcript. Okay, so you are, of course, familiar with antibiotics such as actinomycin, rifampicin, etc. Some of these antibiotics, their mode of action or the mechanism by which they inhibit bacterial growth is inhibition of transcription. So they target the DNA. They target the DNA of these bacteria, and they intercalate. So when we say intercalate, they try to fit themselves into the base pair ladder. Okay, so they insert themselves in between the base pairs in so you have this stretch of DNA molecule. You have the white there are the nitrogenous bases, and the blue, that is the sugar phosphate backbone. So here we see actinomycin B, which has here the planar structure. This planar structure is the one that gets intercalated. So you see there the orange, that is the planar ring of your actinomycin D, and the red ones are the rest of the molecule, which is interacting with the sugar phosphate backbone. Okay, so how does it inhibit transcription in this case? Well, if it's intercalating there and your organism tries to express the gene where the actinomycin D is intercalated, then it can stall the RNA polymerase. Okay? It cannot unwind the DNA if it encounters such structure in the DNA. This is the same mechanism for acridine. Okay, so acridine can also be an intercalating agent. So acridine looks similar to the planar part of your actinomycin D. Another antibiotic is rifampicin or rifampin. This molecule binds the RNA polymerase itself. Okay. And to be more specific, the rifampin actually binds near the active site of your RNA polymerase. So if you can see the rifampin molecule here, it's near the template strand and the RNA transcript that is supposedly being elongated by your RNA polymerase. So the reason is it has this piperazine moiety, which has a similar or analogous structure to your nitrogenous bases. That is why it can fit into the active site of your RNA polymerase, albeit not like exactly how your nitrogenous bases could fit, but it could place itself in the active site of your RNA polymerase. And this way, transcription gets inhibited. Okay, so these are some examples of how your transcription can be inhibited by these molecules. Now, once you have the RNA transcript, it doesn't end there. So after the termination step of transcription, your RNA now will undergo processing. So what you initially get is what we call a primary transcript. So that is the initial mRNA product of transcription. But this primary transcript undergoes several processing before we get the mature mRNA, which is the one that will be translated into a protein molecule. Okay, so we will start with, okay, so I guess we'll just summarize everything first. So there are three general processing being done on your primary mRNA transcript. So the first is the introduction of a 5' cap. Okay, so a guanosine molecule is attached to the free phosphate group at the 5' end of your RNA transcript. And then when you have an mRNA, a primary mRNA product, this mRNA molecule is made up of what we call introns and exons. So there are segments in this RNA molecule that is a sequence for the gene product, but it's how do we say it? It's disturbed, so to speak, by an intron. So the sequence of the mRNA that is supposedly to be translated to the protein molecule is not continuous in the primary mRNA transcript. So you have the exon, but you have the intron. Okay? So the intron is a non-coding end sequence. So it's not part of the sequence that will be translated to a protein product. It's an intervening sequence. Hence, it's called an intron, intervening. Okay? So we shall see later on why this is the case. Okay, there is a purpose why there are introns present in your primary mRNA transcript. Okay, so these introns get removed. Okay, so you cleave, you splice these introns, and the third processing is the addition of a poly A tail at the 3' end of your RNA transcript. So you have a 5' cap and you have a 3' poly A tail. So it's basically a continuous stretch of A's at the end of your mRNA transcript. So it can be as long as 200 bases, or it can be as short as 20 or 80 bases. But you have there a poly A tail. Okay, so the function of your 5' cap and your poly A tail is to protect the mRNA from degradation, because it still needs to be translated, right? So you have to protect it from degradation first. It must be degraded after it gets translated. Hence, you have the 5' cap and the poly A tail. Okay, so let's look first at 5' capping. So 5' capping actually takes place during transcription. Okay, so the cap synthesizing enzyme is found near the C-terminal domain of your RNA polymerase, and this takes place during the elongation step after the C-terminal domain gets phosphorylated. So the cap synthesizing enzyme looks for the 5' end of your mRNA transcript, and it attaches a 7-methylguanosine. Okay, so it's just a sugar with the guanine nitrogenous base there, but this is modified, so you have a methyl group attached to nitrogen 7. Hence, it's 7-methylguanosine. And what's interesting here is that the attachment is via the 5' carbon of the ribose to the triphosphate of the first nucleotide of your mRNA transcript. So we see here a triphosphate linkage. That's your 5' cap. Okay, next we have splicing. Okay, so splicing is the process that removes the introns. And this process is catalyzed, interestingly, by RNA molecules, not by enzymes. So recall that some of your RNAs are actually catalysts, and we call them ribozymes. So ribozymes catalyze splicing of an mRNA transcript. Now, the process is actually classified based on the mechanism. So there are 4 groups of introns, or 4 classification of 4 classes of introns. First is we have the groups 1 and 2 are self-splicing. So you don't need an enzyme for this. Okay? Group 1, a self-splicing group 1 intron, The mechanism is a transesterification reaction, which involves a guanosine residue. No, a guanosine molecule. Okay, so we have a guanosine molecule wherein its 3' OH attacks a phosphate group. So this is at the 3' of an exon because we want to separate the exon from the intron. So this is the 3' of the exon and this is the 5' of the intron. Okay, so the 3' OH of the guanosine attacks the phosphate, so the phosphate group, and then you have there the cleavage of the phosphodiester bond, and now the intron is attached to your guanosine molecule. Okay, so what's happening here is that you have this guanosine molecule that cleaves the phosphodiester bond, attaching the exon from the intron. So after this reaction, the 5' end of your intron is now attached to the guanosine. And then what happens? The 3' end of your exon now has a free alcohol, right? So 2' alcohol. Okay, so the 2' alcohol of the 3' nucleotide of your exon will attack, okay, this phosphodiester bond so that it also gets cleaved, and now you remove the intron, okay, and you connect the two exons. So that is for self-splicing group 1 intron. Now, what about group 2? For group 2, it's still self-splicing, and the difference with group 1 is that you don't need a guanosine here. So the molecule that will attack or will cleave the first phosphodiester bond is found within the intron itself. Okay, so there is an adenine residue found within an intron with this specific sequence, so CAA. So this will be the one that will cleave this phosphodiester bond. So now you form what is called a lariat structure. So this is a lariat structure. And then the free end of your exon can now cleave this phosphodiester bond, releasing the intron, which has formed this structure. And now you connect the two exons. Okay. Now, for the remaining types of introns, they require enzymes. Okay, so they are non-self-splicing. So we will go first with group 3 intron, non-self-splicing, and they are they need what we call a spliceosome. So a spliceosome is an assembly of RNA molecules and proteins that catalyze the splicing of the mRNA transcript. So the spliceosome is made up of a small nuclear RNA protein complexes called SNRPs or SNRNPs, short for small nuclear ribonucleoproteins. Okay, so they catalyze the splicing of group 3 introns. So what's the process? So first we look at the small nuclear RNAs, part of the spliceosome, right? The small nuclear RNAs recognizes sequences in your introns. So for example, for the U1 and U2 snRNAs, they recognize a GU at the start of an intron and an AG at the end of an intron. Okay, so we have two small nuclear RNAs bound at the head of the intron and at the tail of the intron. Then your your SNRPs or your small nuclear ribonucleoproteins will bind. So you see now the U4, U6, U5. So these are the proteins that are part of your spliceosome. So once these small nuclear RNAs recognize their target sequences, the assembly is formed. And another difference here with groups 1 and 2 is that it needs ATP. Okay, so it needs ATP to catalyze the cleavage of the intron from the exon. So a lariat structure is also formed. Okay, and then the intron is released together with the spliceosome assembly or complex. Okay, now this process of splicing can also take place during transcription. Okay, so we have there some spliceosomes interacting with the C-terminal domain. It's not exactly during transcription because this is the termination stage. But the mRNA transcript is not yet released from the RNA polymerase. Now, how do I know that it's the termination stage? Because the CTD is no longer phosphorylated. Okay, so you now have the spliceosome interacting with the C-terminal domain. Notice that you already have the 5' cap there, which is made stable by the CBC protein. So the spliceosome, once it encounters the sequence, so that first sequence at the head of the intron, and then the second sequence at the tail of the intron, it catalyzes the cleavage at two sites, right? And then the intron is spliced off, and then it can repeat the process if it encounters the same sequences. Okay, and once it's done, then the RNA polymerase can release the mRNA transcript. Now, for the group 4, it is also ATP dependent, but it uses an endonuclease. So it doesn't use the SNRPs, okay? So it uses an endonuclease, and this one so far is observed on certain tRNAs. So this non-self-splicing group 4 intron can only be observed so far for tRNAs. So this cleaves at the 5' and 3' ends of the intron, and then it conducts a DNA ligase-like conjoining of the nick in the mRNA transcript. Okay? That's it for the classes of introns. Then last for mRNA processing is the poly A tail. So the poly A tail is also introduced while the mRNA transcript is still bound to the RNA polymerase. So again, we are at the termination stage. Your CTD is no longer phosphorylated. And then we have here the polyadenylation factors, which is essential for the attachment of your poly A tail. What it initially does, what it, no, not attachment of the poly A tail, but the recognition, okay, of the sequence. Okay, so this is called a cleavage signal sequence. So the cleavage signal sequence is recognized by these polyadenylation factors. So once it recognizes that sequence, it cleaves the rest of the RNA transcript. So it's like an excess RNA. It removes that excess RNA. The mRNA transcript is released from the RNA polymerase, and through an ATP-dependent reaction, poly A tail is attached to your cleavage signal sequence. Okay, so the RNA is cleaved by the endonuclease at a point 10 to 30 nucleotides downstream of this sequence AAU-AAA. Okay, so it's cleaved, and then you attach the poly A tail. So the length can be from 80 to 250 nucleotides, beginning at the cleavage site. Again, your 5' cap and your poly A tail serves as a protection of the RNA transcript from degradation. Okay, and last is for eukaryotes. Okay, so here we have 5' cap splicing and poly A tail. This is observed in prokaryotes, but in eukaryotes, we have something special when it comes to the processing of eukaryotic mRNA transcripts. Okay, so wait, this is a summary. So this is not yet for eukaryotic mRNA, but a summary of the processing that can take place. So during transcription, you have 5' capping, you have the extra RNA. Extra RNA gets removed, you remove the introns, and then you have the poly A tail there. And now you have the mature mRNA, which consists only of the exons. Okay, the exons contain coding sequences. Now, the thing is that we have what we call alternate splicing in eukaryotic mRNA. And when we say alternate splicing, it means you can splice a primary mRNA transcript in more than one way. That means even though that mRNA transcript came from one gene, you can actually produce more than one protein product from the same gene. And this is because of alternate splicing. So, alternate splicing is made possible via number one, poly A sites. So there might be more than one poly A sites in your primary transcript. So here you see the two poly A sites labeled. And that means there are two ways by which this primary transcript can be cleaved, right? It can be cleaved at the A1, and then that's where it attaches the poly A tail, or it can be cleaved at A2, and that's where you attach the poly A tail. So you now get two possible mature mRNA transcripts from the same gene. Second is you can have multiple splice sites. So it means the size of your introns can vary. So in this example, you have a single poly A site, but there are two possible 3' splice sites for this intron. So if you have the primary transcript, you cleave at the poly A site and then you attach the poly A tail. Now we can have two splicing that can take place. Okay, one is this is where you cut the intron. So you now have a longer mature mRNA, or you can also cut the intron here and you have a shorter mRNA. But both of these mRNA will give a protein product. So that explains why, although only around 1% of human DNA is made up of coding sequences, and yet we have a lot of proteins inside our bodies. Okay, so the number of genes is not equal to the number of protein products. Okay, so here we see an example of the expression of two different proteins, calcitonin and CGRP, or calcitonin gene-related protein. These two proteins perform different functions in rats. Okay, so this is the primary transcript. You have two poly A sites, and you have 4 introns. Okay, so in the thyroid, that's where calcitonin gets expressed. So in the thyroid, this is how the processing takes place. So the poly A site here is used. So it's shorter, right? And you remove the introns. So now you have a short mature mRNA. You translate this, you get calcitonin. But in the brain, for the same gene, it's processed differently. So it uses the farther poly A site. So yeah, it's cleaved there, and you have the poly A tail. And now you remove all of those intervening sequences, or the introns. Now the intron this is the whole intron. So it includes the sequence for calcitonin. So this is an intron. This is the second intron, and this is the third intron. So that's the one that is removed as well. So you have a bit longer mature mRNA. If it gets translated, the protein is CGRP. Okay, so that is what we call alternate splicing in eukaryotic mRNA.